Ma'am, I can't hear you.
I Hello and welcome to Excelsior Academy. So I wasn't saying that time anything. Okay, so in the initial lecture, we have discussed about the different types of cell lysis, right? If you remember the cell lysis, we have talked about if it is bacteria, then what should be the method? If it is animal cell, then what should be the method? And what about the plant? Okay, so in the case of plant and yeast, the cell wall is big. So you might need to add even enzymes also. Okay, so we have discussed those. Okay, so if I'm asking you to carry out the animal protein purification. So what will be your first step then? Yes, for animal, what will be your first step? If you have got animal cell lines, okay, I'm talking about cell lines, I have not taken the tissue. So what is your first step if you are targeting protein? And getting protein is easy as compared to the other things. Yes. So what will be the first step, Tanvi? Shruti?
I wasn't there, ma'am.
Acha, you were not there. Okay. What about Tanvi?
मैम एनिमल
सबसे पहला स्टेप क्या होगा? So there is a plate I have given you. Okay, in that you have got that pink color ka media, which is your RPMI or DMEM. Okay, so वो आपका pink color का media रहता है. You have got the cell lines over here in the culture plate. ठीक है? Now what you do is first you remove the media. I have shown the video also of this. So first you remove the media. Okay. Then next step is you wash the cells with the help of your 1x PBS. What is PBS? It is phosphate buffer saline. Okay, so it has got the the pH and the constitution which is similar to your अपना serum. ठीक है? Not serum, the blood circulation pH and the osmolarity. Sorry. So the osmolarity and the pH is similar to our blood circulation system. Okay, so what you do is then 1x PB से आप उसको wash करोगे. Till then, the thing is that on the culture plate, your cells are attached over here, ऐसे. Okay, your animal cells. Then what you do is you add trypsin. Okay, now trypsin, what they are going to do? They are going to chop off the proteins which are helping the cell in order to adhere to the cell. Okay, so then what will happen? The adherence will go. And next, what you can do is in the PBS itself, you can have the suspension of the animal cells. Now, what you can do is either you can go for first thing is homogenization. Okay, you can do that. Or second, what you can do is you can vortex. Okay, you can vortex, and then after that you can centrifuge. So whenever we make it like this, it means it is centrifuge. Okay, now if you centrifuge at high speed, what will happen? Because of the shear force, what will happen? The centrifugal force, the membrane will rupture, which was not ruptured during the vortex, will rupture during the centrifugal force. And at the end of your test tube, you are going to get the cell debris. Okay, and on the top I can get the protein. Then I can start my experiment. Okay, simple. If anyone is asking how you are going to start or going to get the protein from particular sample, these are the steps. Okay, so we have discussed about the cell lysis in the previous lecture. Now let's discuss about the enzymatic and the purification of DNA and the last quantification of DNA. Now coming to the enzymatic treatment, already we have discussed also. So what are the enzymatic treatment? Earlier what we have discussed, so we have discussed that in the case of the bacteria, cell wall rupture, you need lysozyme. Okay. Now, there was this question when one of the scientists was asking that we know that we use lysozyme in order to, to disrupt the cell wall of the bacteria and all. And we also know that in our tears also, the concentration of lysozyme is very high. Can you relate these two and justify? Yes, anyone? I guess the question is clear. Why does the tears, human tears, have got high concentration of lysozyme enzyme present?
They are a natural antibacterial agent. They protect the eyes.
Yes. So, see, we don't have any other ways by which you can protect your eyes. First of all, either you can close your eyes. That is like physically you can protect your eyes, right? Closing of the eyes. Now, what if there is an infection? Okay, so every time you cannot put all these antibiotics, the chemicals, that is the last stage when nothing is working. So as you said, yes, it is correct. It is a natural way of protection from the bacteria. So that's why we have got high concentration of lysozyme. क्योंकि ये expose होते रहते हैं, आपकी आंखें हमेशा बाहर की तरफ expose रहती हैं, right? And you will also see that if you know life में कभी रोए हो and आपने आंसू पिए हैं, okay, literally अगर किया है, I'm not going with the dialogue once. So you must have noticed that the salt content is also present in the tears. Namkeen rehte hain aansu. Correct. So this is also, if you see the concentration of salt or the osmolarity is also very high. Again, that is by the changing the osmolarity, it is making sure that the bacterial cells are not able to survive in those conditions. So if you see the tears has got high concentration of lysozyme, high concentration of salt, and it makes sure that the daily में हमें eye infection ना हो करके, ठीक है? Later on we also get and all, that is different scenario. Okay. Now in the case of plant, so this also depends like what region of the plant you really are focused in getting the DNA or the protein or the carbohydrate. Okay? So we have got the pectinases enzyme. So you know it is to degrade the pectin. Then you have got cellulases for cellulose. Then hemicellulase for hemicellulose. Correct? And then we have got the amylase wherever the high starch content is present. Okay. Now, in the case of animal, as we discussed, it is trypsin. So, few cases trypsin is there. We use chymotrypsin also. Okay. So, these are the proteins which we use for animal cell. Now, there are other proteins also. So, removal of RNA from the DNA preparation is easily achieved by the treatment with ribonuclease A. RNase A. Okay. Now, if you see this thing, very important. Okay, this slide is very important to understand whenever we are doing with the DNA RNA purification. ठीक है? अभी आपको पता चलेगा क्यों. So let's see. So we know that अगर मैं DNA prepare कर रही हूँ, so I don't want the contamination of RNA. So RNA can be removed with the help of the RNase. So if you remember, whenever we go for, you get the supernatant and everything. Okay? So what I can do is I can add proteases, RNases, so they can degrade the protein and the RNA. Correct? They will become the ribonucleotides or amino acids, and they will be very light in solution. And हम next round of centrifugation में हम इसको निकाल सकते हैं. Now, since RNase is a very heat stable enzyme, okay, it is a thermostable enzyme. It is easy to ensure that it is free of all traces of DNases that would otherwise degrade your DNA simply by heating the enzyme before use. Okay? इतना थोड़ा समझ में आया? See, अगर मैं heat कर रही हूँ sample, I know DNA is stable. See, यहाँ पे ना बहुत ही interesting है. So you have got RNA, you have got DNA. ठीक है? RNA is thermostable. Okay, but RNA is not stable. Here, your DNA is stable, but DNA is not. Are you getting the point? ये समझ में आ रहा है? See, evolutionary, how beautifully they have maintained. DNA आपका stable है. मैं यहाँ पे heat की बात कर रही हूँ, ठीक है? I'm talking about heat over here. Your DNA is stable at high temperature. Okay. When I say high temperature, it doesn't mean that DNA means stable means it won't be broken. Okay. Double strand break or single strand break नहीं होगा. There will be a denaturation. That is like double strand से आपका single strand जरूर बनेगा, but there won't be any breakage, okay, at a certain point. Now, if I just slightly increase the temperature, the enzyme DNase is not stable. So what will happen? By heating the sample, I can ensure that DNA is present without DNase. So now DNase is not able to break your DNA. Okay, so my sample is now free of DNase. Is this much clear, guys? Are you able to follow? थोड़ा सा confusion हो सकता है. अभी तक clear है श्रुति and Tanvi?
Yes, ma'am.
Okay, now let's talk about the second scenario. Okay, now RNA is not stable. जरा सा भी heat रहा और fluctuation रहा, RNA once it is out of the cell, it is not stable. Okay, so of course at slight temperature it is not stable, but RNase जो enzyme है वो आपका thermostable है. Now when we do DNA purification जब आप करते हो, it is very easy. Okay, you can just randomly without wearing gloves, you can start doing the DNA purification, and beautiful bands you will get at the end of your agarose gel electrophoresis. But in the case of RNA, it is not like that. Your hands, your fingertips, okay, every time there is a secretion of RNase A on your hands. ये जो अपना जो skin है, we always say that, right? They are shedding the cells every day, the palm and the foot region जो आपका रहता है, right? So on the palm, always we are having RNase A. So जब भी आप कुछ भी RNA related purification करते हैं, you need to make sure that everything is sterilized or alcohol clean. आपके हाथ भी. And every step, agar suppose I have taken the tissue sample, before that I should alcohol wiped my hand, and of course I should be wearing gloves. Without gloves, if you try doing RNA purification, never ever you're gonna get. And that too, mRNA is very very unstable. Okay, so जैसे मैंने sample लिया, मैंने crush किया, I started doing those phenol chloroform purification of RNA, जो भी आपका protocol है. Where आजकल तो खैर lab में there is this trizol reagent, आपने सुना होगा trizol. If you have done, so kit आता है अभी तो company से directly just add the sample to that. After just crushing the sample, add the trizol reagent and do 2-3 times centrifugation, and you get the mRNA. And immediately after mRNA, we go for cDNA preparation, complementary DNA preparation. Why? Because mRNA is not stable. तो हमको क्या करेंगे? Complementary DNA जल्दी बना देंगे using RT, reverse transcriptase. Okay, we'll come to that. Don't worry, we'll come to that. So the point is that here every time you have to be very careful when you're doing RNA preparation. DNA is simple. Okay. Now protein contamination can be removed by the enzymes like proteolytic enzymes like proteinase K हो गया है, proteases हो गए हैं. So many are there by which you can remove the protein contamination. Okay, is this slide clear to both of you? This is actually important to understand. Okay.
Yes, ma'am.
Yes,
Now,
ma'am.
okay. So these treatments are applied if necessary in different nucleic acid purification protocols. See, अगर आप RNA कर रहे हो, of course आप RNAs नहीं ऐड करोगे, वहां पे आप DNAs ऐड करोगे. DNA प्यूरिफाई कर रहे हो तो RNAs ऐड करोगे. प्रोटीन दोनों केस में आप प्रोटीएजेस या प्रोटीनेज के ऐड कर सकते हो, because you don't want protein contamination. Now, when you are talking about protein purification, then of course you need to add protease inhibitor. Okay, there are certain things which is not mentioned in the slides. Please note down. So अगर आप protein purification कर रहे हो, so you need protein inhibitor. Have you ever heard of PMSF? PMS सुना है क्या कभी?
Yes, ma'am.
Full form बता सकता है कोई? It is important. Full form क्या होगा इसका? So, okay, at least you know that this is your protein inhibitor. Oh, sorry, protease inhibitor. Okay, so it will stop protein degradation, and this is how you can purify. Ab see, यहाँ पे we don't add DNases and RNases. Can you tell me? First of all, it's a protein, so it's an enzyme, right? So we won't add additional DNases and RNAs in the protein purification step. Can you just justify by 2 statements? क्यों नहीं डालते? सोचो, क्यों नहीं डालते? क्या रीजन होगा? जब हम डीएनए प्यूरिफाई करते हैं हम आरएनए डालते हैं, प्रोटीएज डालते हैं, ताकि we don't get the contamination from RNA and protein. Similarly for RNA we add DNases and proteases, but during protein purification we don't add DNases and RNases. ऐसा क्यों कर रहे हैं हम? Are you guys thinking? Question is clear?
Yes, ma'am.
Okay, सोचो. पूरा 1 घंटा मत लेना, नहीं आ रहा है तो प्लीज टेल मी. हाँ, but I really want you guys to think. Okay, any guesses? It's okay if it is wrong, it's okay. We are here to learn only. हाँ, तन्वी आ रहा है आंसर? क्या होगा? This is your phenyl methyl sulfonyl fluoride. Okay, phenyl methyl sulfonyl fluoride. Okay, चलो ठीक है, मैं आंसर देती हूँ. सिंपल आंसर है, बहुत ही सिंपल था. सिंपल सी बात है. यू आर डूइंग प्रोटीन प्यूरिफिकेशन. Add addition of DNAs and RNases. This is again a protein by nature. तो मैं प्यूरिफाई कर रही हूँ मेरे कंटेंट का, like whatever the protein I am interested. So if I add additional, you are increasing my load of purification. Is this point is clear?
Yes, ma'am.
Second point क्या हुआ? See, we are doing so many times protein purification and हम उसमें centrifugation करते हैं. So we know that proteins are lighter in density as compared to DNA and RNA. तो जब हम हाई सेंट्रीफ्यूगेशन फोर्स करते हैं, जैसे कि हम 20,000 ने स्पिन किया, आरपीएम किया या जी किया, सो व्हाट विल हैपन? बिकॉज ऑफ द डेंसिटी डिफरेंस, so DNA RNA are heavier as compared to the protein. So what will happen in the centrifuge? In the bottom they will be forming the pellet. So supernatant ले लूंगी. So this is going to have only the proteins then. Okay, so मुझे additional DNAs RNAs add करने की जरूरत नहीं है. So I can just centrifuge my sample and remove the DNA. Is this much clear?
Ma'am, can you repeat the second point?
Okay. One second. So I was saying, first point is clear, right? So we don't have to add additional so that the protein purification load will increase. So I'm not adding DNAs and RNAs because they themselves are protein by nature. Now second is the density. Okay, it is the density. So density means, simple सी बात है ना, DNA is going to be very heavier, RNA is going to be heavier because of the backbone. इसमें phosphate है, इसमें sugar है, इसमें bases हैं आपके. Whereas in the case of proteins only amino acids. So if you see the weight wise, okay, and you also know इतना बड़े DNA में आपके छोटे छोटे protein होते हैं. Okay, so the density, the heaviness of DNA and RNA is more as compared to your proteins. So when I am going to centrifuge, okay, centrifuge जब मैं कर रही हूँ at high level, जैसे suppose let us say for example 20,000 RPM हो गया या 50,000 RPM हो गया, okay. So what will happen? Because of the centrifugal force and the density difference in the centrifuge tube, at the bottom, this pellet, I am going to have DNA and RNA, okay. And supernatant, we are going to have only protein. अभी clear है? Protein. अभी clear है?
Yes, ma'am.
अब क्या है, see if my target is to purify proteins, अब मुझे super nutrient तो मिल गया, now I'm having the thousands and different types of proteins are there. Now next step should be like what kind of protein I should have. मेरा प्रोटीन बेसिक है या एसिडिक है, सो आयन एक्सचेंज करूंगी. अगर एफिनिटी क्रोमेटोग्राफी करना है तो मेरे प्रोटीन में कुछ टैग होना चाहिए. अगर मेरा प्रोटीन बहुत हैवी है, लेट अस से दैट माय प्रोटीन इज अराउंड 2000 किलो डाल्टन, ओके, मैं 2000 किलो डाल्टन बोल रही हूँ, सो आई कैन गो फॉर जेल फिल्ट्रेशन क्रोमेटोग्राफी. So based on the size of the protein, you can have different ways of protein purification, which we'll cover later. So we have got different chromatography techniques. You must have heard of affinity ho gaya, ion exchange ho gaya. In ion exchange, you have got anion exchange and cation exchange is there. Then affinity ho gaya. Okay, then size exclusion is there. Hydrophobic chromatography is there. So many chromatography techniques are there now. Okay. So based on your protein requirement and the size and charge of the protein, you can go for the purification. So we'll cover this later once we complete this. Okay, so this much is clear. Okay, so however, in some protocols, they are omitted either because the contamination is unimportant for specific purpose or the contamination will be removed anyway throughout the subsequent steps as described. तो जैसे मैंने बोला, हम अगर प्रोटीन कर रहे हैं, हम जितने बस सेंट्रीफ्यूज करेंगे, जितने भी heavy particles होंगे of DNA and RNA, they will be removed in the subsequent steps. Okay, now coming to the third category, that is your purification of DNA and RNA. अभी क्लियर हो गया, enzymatic steps clear है. So we have seen that there are different types of cell lysis, enzymatic treatments are there, what kind of different enzymes we use. Okay, now let's come to the main part, that is purification of DNA and RNA. So first, obviously, we'll talk about DNA first. Okay, so the first step is your phenol chloroform extraction. Okay, so removal of protein is particularly important because we have got the enzymes and all which can degrade nucleic acid. इससे सबसे पहले हम proteases ये सब हटा देंगे. And the first step what we are going to use is the liquefied phenol or the mixture of phenol and chloroform. So ये depend करता है आपकी कंपनी ने कौन सा वाला आपको दिया है, or you can go for the conventional method also. Now one thing is that before I start with phenol and chloroform method, you should know that the phenol comes in powder form. Okay, it is in the pinkish, light pinkish powder form में रहता है, and it is highly dangerous. It means what? At room temperature, जहां से भी थोड़ा सा गर्म हुआ, okay, हल्का सा हीट भी उसको मिला, it can just burst out. अगर वो किसी कंटेनर में है, it will just burst out like a bomb. आपका वो स्किन जला देगा. So you know that if you touch phenol, अगर phenol आपकी बॉडी में गिरता है, it burns that area. Okay, it burns that area. So what you can do is, in order to use phenol for proper this reference, okay, you make it as tris saturated phenol. Guys, सुना हुआ लग रहा है? लैब में किया है आपने कभी? नहीं, none of you? Shruti, Tanvi, you have not used, not done, और या डेमो देखा होगा?
Yes, ma'am. Demo दिखा है.
हाँ, so we have seen उसमें हम क्या करते हैं, हम phenol and chloroform यूज करते हैं. तो फर्स्ट स्टेप में क्या होगा, आपके अनवांटेड प्रोटीन्स चले जाएंगे. ठीक है ना? When you say phenol and chloroform, so when you say tris saturated phenol, okay, this is the first step actually. So tris saturated phenol means you are bringing phenol to this particular pH. pH यहाँ पे 8 रहेगा. ये थोड़ा सा क्या है ना, आपका practical level का knowledge है. Okay, and this is like universal. If you see, it is universal. यानी कि बाहर भी यही pH पे काम होता है. Everywhere this is a fixed protocol. Okay. So when you say tris saturated phenol, इसका मतलब क्या है? You are making it liquefied, right? As I said, it is a pinkish powder. तो आप weigh करोगे. Be careful whenever you use phenol, because there was this accident at one of my lab. Like when I joined, and before that, okay, before मेरे joining से 2 साल पहले की बात है. So I did my PhD. I started in 2010. So 2008 की बात है. So this girl, she kept the phenol का bottle on the you have got this burning stand रहता है ना, थोड़ा सा उसको हीट करके लिक्विड बनाने के लिए, तो यहाँ पे आप फ्लेम रखते हो. So in the fume hood, she kept this phenol का bottle like this, but she forgot to open the cap of the phenol bottle. So when you heat it, what happened? वो पूरा फ्लेम्स उसका जो रहता है, फ्यूम्स वो इकट्ठा होता रहा एंड व्हेन शी जस्ट केम एंड शी जस्ट टच द लिड एंड इट बर्स्ट, तो पूरा फिनॉल उसके ऊपर गिर गया एंड इट वाज रियली वेरी डिफिकल्ट, इट वाज अ वेरी सैड, इट वाज रियली बैड एक्सीडेंट. एनीवेज, so this says that phenol you have to be very careful, but आजकल कंपनी आपको saturated phenol देती है, so it's okay. Okay. हाँ, so now what is going to happen is phenol and chloroform, they are not immiscible with water. What does it mean immiscible? मतलब water से mix नहीं होगा, right? So because of this you are going to have two layers when it is added to the cell extract. अभी आपने क्या किया, जो भी आपने क्रश किया, सेंट्रीफ्यूज किया, okay, whatever supernatant you got, now to that you have added your phenol and chloroform and it is not going to be mixed with the water. So अब क्या है, whatever is your water soluble, okay, it will come into the water layer, और बाकी जो आपका हाइड्रोफोबिक है वो आपका सारा phenol chloroform layer में आ जाएगा. So you are always going to have the two layers. Is this much clear? Yes.
Yes, ma'am.
Okay. Now, when the mixture is mixed or agitated, the proteins will be denatured and precipitated at the interface. अब यहाँ पे देखो, मैंने इसको न्यूट्रल पीएच किया है, ओके, एज आई सेड, अभी मैंने बोला अगर मैं इसको ट्रिस सैचुरेटेड फिनॉल बनाती हूँ, so that pH is going to be around 8. I mean, neutral is 7, I know, but for the buffer range, it is 8. ठीक है? Now, what you can see, if it is, I'm using tris saturated pH 8.0 वाला, you can see that here I've got the phenol layer, okay? यहाँ पे आप देख सकते हो एक प्रोटीन प्रेसिपिटेटेड लेयर आएगा, then aqueous layer जहाँ पे आपका DNA and RNA is soluble, okay? So you know that the proteins are also soluble in water, but not all proteins, right? But when I add phenol, what happens? All the proteins are tend to form aggregates, so they will get precipitated out, and यहाँ पे आपको एक white color की layer दिखेगी. Clear? थोड़ा सा clear है? हाँ? So this is your append जो रहता है आपका 1.5 ml का. So in that you just add your sample, add phenol. Now one more thing, अगर आपने 1 ml sample लिया है, so then you should add 1 ml of phenol. किसी protocol में 0.5 ml phenol रहेगा, but depending on that, like whatever protocol you are following. Okay? Clear guys? ये थोड़ा याद रखना पड़ेगा. This figure you need to remember that when you do phenol extraction, the top layer of aqueous layer is having your DNA and RNA, then protein precipitated in the phenol. Now tell me, Shruti, which layer are you going to take for the next step?
The middle layer.
We are doing purification of DNA and RNA. So which layer you should be taking?
The top.
top. Okay. See, once you have added phenol, your protein has precipitated. Now, protein precipitation means what? It is denatured. वो किसी काम का नहीं रहा है. So you can't think of having a protein purification step once the phenol has been added. ठीक है? अभी यहाँ पे भी ध्यान दो. If you're using an acidic pH वाला phenol, then the aqueous layer only RNA is there, protein precipitate रहेगा यहाँ पे, and then phenol layer DNA is there, which later becomes difficult to remove the phenol, okay, because phenol will also give reading at 260 nanometer, so that becomes a problem. So इसलिए हम क्या करते हैं, instead of having an acidic pH वाला, we go with the neutral pH वाला, so that when you quantify your DNA, you are sure that the phenol has been removed. Okay, so अभी हम क्या करेंगे next? I'm gonna take only this layer. ये ऐसे दिखेगा, एकदम white transparent दिखता है. You can see this as white in color, और ये थोड़ा turbid pink color दिखेगा. Phenol का जो color रहता है वो थोड़ा pinkish रहता है, ठीक है? So I'm gonna do is I'm gonna take this top layer into another append-off. ठीक है? ये आपको थोड़ा याद रखना ही है, whatever happens. Okay. Now coming to the next. So if you're using equilibrated बोलते हैं, ठीक है? अगर आपने उसको buffer के साथ किया है, tris हम लोग usually we use. So it will remain in the aqueous layer. If you carry out with the acidic phenol, then DNA will partition into the organic phase, यानी कि वो phenol phase में रहेगा, ठीक है? So phenol is naturally acidic. So you equilibrate with the water or the buffer to get the appropriate condition. So this is clear. I have already mentioned. Now it is also this phenol extraction is also useful in the subsequent stages of manipulation when it is necessary to ensure that all traces of enzymes have been removed. Now coming to the alcohol precipitation. Okay. So following phenol extraction, you will have a protein-free sample of your nucleic acids. Okay. Now what is you are going to do is, फिर भी उसमें कुछ traces of phenol and chloroform will be there when you do. पहले हम क्या करते हैं, फिनॉल ऐड करते हैं, सेकंड स्टेप में हम क्लोरोफॉर्म ऐड करते हैं, सेम यू गेट, एंड देन थर्ड स्टेप में हम एब्सोल्यूट अल्कोहल ऐड करेंगे. ठीक है. नाउ phenol in particular does not have any significant degree of solubility in water, and this could lead to denaturation of enzymes in the further steps if I am working on. So now I'm going to do is precipitating the DNA with the help of the alcohol. So either I am going to use isopropanol or the absolute alcohol, but हम जहाँ तक मैंने भी experiment किए हैं, we have also always use the isopropanol, and we make sure that isopropanol is kept in the fridge. तो जो ठंडा रहता है तब भी use. Okay, so far so good, guys. Are you able to follow?
Yes, ma'am.
हाँ? Don't worry, we'll
Yes.
summarize also. ठीक है, हम इसको summarize भी करेंगे कि कैसे क्या हमने किया, क्यों करते हैं, वो important है अभी. बाद में तो you can remember, not a problem. Okay, so now you know why we use phenol, but you know why we use chloroform and isopropanol now. Okay. Now, so in the presence of monovalent cations, nucleic acid precipitate forms which can be collected at the bottom of the test tube by centrifugation. See, अभी हमने क्या देखा that in the few steps of the phenol and chloroform, you were taking the top layer. ठीक है. अब आपने क्या किया है, you have added the alcohol. I'll write alcohol, but keep in mind it is always the isopropanol. Okay, cold isopropanol. Now, once you have taken the isopropanol, now what will happen is your nucleic acid is going to get precipitated. Now, once it is precipitated, it is going to come at the bottom. ठीक है. So, इस step में you are going to remove the supernatant and you are going to collect the alcohol. Is it clear? Clear है? ये आपका याद रखना जरूरी, बिकॉज जब भी हम आगे ऐसे क्वेश्चंस आते हैं या इंटरव्यू में पूछते हैं, उससे यही पूछा जाएगा कि अगर मैंने अल्कोहल स्टेप कर रही हूँ तो मैं पेलेट लूंगी या सुपरनेटेड लूंगी. ओके, सो ऐसे आपको कंफ्यूजन आ सकता है. ओके, क्लियर? ओके, लेट्स गो टू द नेक्स्ट स्टेप. ना, so you have got the DNA solution. मैंने भी इसमें इथेनॉल डाला. Of course the sample will increase. फिर हम इसको centrifuge करेंगे. Now can you see DNA precipitate ऐसे. Now frankly speaking it is not visible. Okay, हमें दिखाई नहीं देता है DNA unless until आप plant से कर रहे हो. So you can see a white dot. Okay, ऐसे आपका appendix रहता है, यहाँ पे छोटा सा एक ऐसे छोटा सा white dot दिखता है. In the case of the plant, अगर tissue sample ले रहे हो, okay, if you have taken animal tissue sample, then also you can see a small dot. But अगर bacterial कर रहे हो या animal से कर रहे हो, sometimes you just can't see anything. ऐसा लगता है जैसे कुछ नहीं है. So is this much clear? So ethanol step means after this step you are going to take the pellet, okay, जो आपका present होगा. Clear, guys? Okay. मैं पूछूंगी हाँ, so don't get distracted. Okay, so now this is different little. So if I go back, so now what you're going to get is DNA precipitate. अब मैं इसको क्या करूंगी? There is like TE buffer, TRIS का buffer रहेगा आपका. Okay, so TE buffer रहता है. There are certain TAE buffer रहता है. What you can do is, or you can have milliQ. So milliQ means double deionized water. It is your double deionized water. यानी कि आपने सारे ions मिल सब हटा दिए हैं. Okay, and in that around 20 microliters, 20 से लेके 50 microliter तक you can add. Okay, इसमें मैं add कर दूंगी and I'll reconstitute the DNA. Now my DNA is ready. So this is how you are going to do the DNA purification. Okay, अब अभी तक क्लियर है गाइस?
Ma'am, can we summarize it once?
ओके, मुझे लगा ही था थोड़ा सा कंफ्यूजन आ गया होगा. हाँ, ओके. सो ओके, टीई भी थोड़ा सा याद रखो. दिस इज योर ट्रिस ईडीटीए बफर. ठीक है, ट्रिस ईडीटीए. Okay, so you in the lab when you join, you won't be provided with the kit directly. You will be asked to do the conventional method, and after that, once you become pro in your conventional method, you can go with the kit. Okay, because kit is costly and it is limited. Like it is possible only for the 100 samples. So आपको practice कराने के लिए हम ऐसे kit नहीं देते हैं. So you will be taught like centrifuge कैसे करना है, appendix कैसे handle करना है. So once you're good in handling all these things, okay, then the kit will be given, because kit is like once used it's gone. और आपको अगर सैंपल नहीं आया तो फिर then it's a wastage of money, and all these kits are very costly. Okay, so let's summarize. So is this much is clear ki if suppose let's talk about the plant DNA. Okay, let's talk about the plant DNA. So you have taken plant leaf. So you know that what you have to do. First, take the weight of the leaf. Let us say that we have taken 100 gram of the leaf sample. I have used liquid nitrogen in order to make it brittle so that it becomes easy for me to crush in mortar pestle. Okay, itna clear hai? Ab mortar pestle mein it is hard. Now with that I'm able to make it a fine powder. Okay, now fine powder is made. To that I will add buffer. Okay, so that they become reconstituted. आपका DNA, protein, carbohydrate, phenol जो भी इसमें present होगा सब. Now I'm going to centrifuge. So what will happen in the centrifuge? Only the cell debris, okay, only the cell debris which are present will be removed. Guys, stop me अगर कोई step समझ में नहीं आ रहा है, ठीक है? Now I am going to take the supernatant. Now this is ready, my cell lysate, okay, for my DNA purification, it is ready. Now to this supernatant, if it is 1 ml, I'm going to add 1 ml of my phenol, tris saturated phenol. मैंने add कर दिया, okay. अब मैंने क्या किया, add किया. We do this ऐसे appendix को लेके we make it ऐसे up and down so that it gets mixed properly. We keep it for some time and again centrifuge. अब centrifuge किया तो क्या हुआ? I am going to get in my appendix 3 layers. First, यहाँ पे आपका phenol रहेगा pink color का. इसके टॉप पे you will have protein precipitate जो अब मेरे काम का नहीं है, which is in white color. ठीक है? Then on this top I'm going to have my aqueous solution जिसमें मेरा DNA and RNA है. Okay? Now, if you are not interested in RNA, then हम पहले step में supernatant में ही हम RNAs add कर सकते हैं so that I can remove the RNA. So let us say that I have added RNAs and remove the RNA part. Okay, तो अब मेरे पास RNA नहीं है. I'm just focusing on my DNA. Is this much clear? मैंने phenol कर लिया है. अब मैं chloroform कर रही हूँ so that I can remove the phenol and the बचा हुआ protein. Again, what I'm gonna get is these 3 layers. And again, the supernatant I'm going to take. Okay. So in my third step, I'm having my DNA solution. This is what I'm gonna get after my phenol chloroform step. Clear? Itna clear hai? To this, I'm adding my cold alcohol. यहाँ पे ethanol है या कुछ भी हो. Ethanol हम add कर सकते हैं. हम isopropanol use कर सकते हैं. But people have shown that with experience, if you use isopropanol, it gives better. But we can use ethanol also. Absolute ethanol, okay? No addition of water. So मैंने मेरे DNA solution में मैंने add किया मेरा ethanol, okay? Centrifuge किया at high speed. What I'm gonna get now? The DNA will precipitate here. और जो भी salt उसमें जो present होगा वो सारा आपका remove हो जाएगा, okay? So now at the bottom I'm gonna have the DNA. To this DNA I'm going to add TE buffer, which is your tris EDTA buffer. Tris EDTA buffer, and you know EDTA का क्या काम है? It's a chelating agent, right? So जो भी excess salt present है, अगर आपका calcium present है, zinc या magnesium present है, which should not be present in the DNA sample, so that will be taken out in the EDTA. Okay. Now I'm going to add around 20 to 50 microliter of the sample of the TE buffer to this Eppendorf, and I'll reconstitute. So now I have a DNA, purified DNA solution. अब clear है?
Yes, ma'am.
एक बार और सुनोगे तो हो जाएगा. I am sure this is sufficient. This is how you do in the lab. अगर आप अभी नेट पे सर्च करोगे कि कैसे डीएनए आइसोलेशन करते हैं, क्या करते हैं, तो आई गेस मैक्सिमम टाइम व्हाट यू गोना गेट इज हाउ द किट इज वर्किंग. ओके, सो विल डू दैट आल्सो, बिकॉज आजकल द डीएनए प्यूरिफिकेशन बहुत ही सिंपल हो गया है, एंड जो भी स्टेप मैंने अभी आपको बताया इट विल टेक एंटायर डे. ओके, क्योंकि आपको ऐसे 10 मिनट तक हाथ से ऊपर नीचे करना पड़ता है, फिर 15 मिनट उसको टेबल पे छोड़ना पड़ता है, देन उसके बाद 20 मिनट आपको सेंट्रीफ्यूज करना पड़ता है. सो एक एक स्टेप इज गोइंग टू टेक अ टाइम. अब क्या है, व्हेन यू यूज ऐसे किट, so they have used small ऐसे chromatographic columns into which the DNA will bind, and आपको ये सब करने की जरूरत ही नहीं है. So that also I have to discuss because आजकल आता है क्वेश्चन उस पे भी. ठीक है, so yeah, so we'll discuss that. So far so good guys. Then I'll be stopping. मैंने actually one second.